Good morning, everybody. This is the third day of the Women in Bioinformatics and Data Science Latin America. And this session will be about genomics and evolution. And we have Dr. Ana Tereza de Vasconcelos as a keynote speaker. And before I especially Lucy, Anna, Christina, Georgina, and all the people that are helping them. Thank you so much. The meeting is great, and it's very difficult to organize a meeting online in the middle of this pandemic, and it's been super. Congratulations, girls, you are fantastic. And the next slide. Okay, no, it's not working. Sorry. I have to stop sharing because the slide's not working. Just a minute. Now it's working. Then Dr. Ana Tereza, it's a friend, a good friend of mine for a long time. And I met her when she was the principal organizer of the Genomics Network in Brazil. She's a biologist and has a master in biophysics and PhD in genetics. She is actually full researcher at the National Laboratory of Scientific Computation, LNCC, in Petrópolis, Brazil. And she's also professor in the Department of Genetics at Federal University of Rio de Janeiro. She participated in the creation and she was the first president of the Brazilian Association of Bioinformatics and Computational Biology. And she is the coordinator of the Computational Genomics Unit, Darcy Fontoura de Almeida at the LNCC, and also the coordinator of International Associate Laboratory from CNRS. She has experience in the area of bioinformatics and computational biology, working mainly on genomics, development of mathematical methods, and computational tools applied to genomics and genome annotation. She's one of the most regarded bioinformatician in Brazil. She's a reference for bioinformatics in Brazil. Thank you, Ana Tereza, for being here and accepting our invitation to be the keynote speaker of this morning.
So, Gloria, thank you very much to introduce me. It's a great pleasure to be here with a lot of women, girls. It's a fantastic idea. It's great. It's important to include women in most of the conference. I have been for a long time invited to in a conference in America, in Latin America, because we are not able to find a lot of women to talk about bioinformatics and genomics. Yes, Gloria told us, I'm starting bioinformatics a long time ago, and for that reason I'm a reference in Brazil because I'm oldest, Gloria. That's the only reason that it's happening.
This is not true, Anna.
Look my hair. So as Gloria introduced me, I'm working in the National Laboratory for Scientific Computation. It's one of the Institute for Research from the Ministry of Science and Technology in Brazil. And my main area is bioinformatics. And today I will talk briefly about some projects that we are performing in the lab, working from the virus to the human genomes using the bioinformatic methodology to analyze this data. And thank you again for inviting me to be here. Oh, it's not working. I will stop to share my... Oh. Now you can see.
Yeah.
Yeah.
Yeah, we can.
But
Let's try.
But
Okay.
okay,
Yeah.
now it's working.
Yeah, it's working.
Yes.
So yes, sorry about that. So at LNCC, we have a facility for genomics. So we start with the 454 a long time ago, and now we work most of the time with the Illumina platform, but we will also have another one. And one of the things that's important at LNCC, that we have a supercomputer, that's the name is Santos Dumont. That's the biggest HPC supercomputer in Latin America. So we can go through generate the sequence and then analyze the sequence in the same place. Oh, it's not. Oh. And so in the lab, we are working with a lot of genomes from bacteria, virus, exome, metagenomes, and transcriptome. And then we analyze using the Santos Dumont supercomputing. And then we develop many tools to analyze, to visualize, to integrate this data, and to promote the people to using our source. So the name of the main software that we have been developed is Sabia. It's an hosted. We start with bacteria in the 25 years ago. And then we moved to develop another tools to analyze the new data, such as the transcriptome, when we start to work with that, exome, virus, and metagenomics. So talking a little bit what we have been doing the last year, we start to work with virus since they started the outbreak of Zika in Brazil. So the Zika virus arrived, the initial detection was in 2015, but I think that the outbreak came from the Pacific Island, introduced to Brazil in 2015, and then spread for America. The Zika virus is a flavivirus. This is a single positive RNA virus with about 11 kb, and there is 3 main proteins and many others non-structural proteins. And in the beginning, we worked with the Fiocruz, the Instituto Oswaldo Cruz Fiocruz in Brazil, and we are able to sequence in the genome from the urine saliva that has been infected in viral cells. We can see here the cytopathic effect when we infected the cerebrals. We are able to recover the genomes from these samples, and then we sequencing these genomes and align it with the other strains spread all over the country. And we have published this work in Brazil in 2015. This is very important because at this time we didn't know if we can transmit the virus, the Zika virus, in saliva. And at this time, in Brazil is the time from the carnival. So we announced that we can transmit in acute phase the virus, the Zika virus, one day before the carnival. So it's a very important announcement that caused a lot of disturbance in Brazil for that reason. And we also work with a lot of bacterial genomes related to resistance, to different resistance in Brazil. We have different bacteria that are spread all over the country, and they're very important problems for the system health in Brazil. One of them is Acinetobacter, another one is the Klebsiella, and also Pseudomonas. So most of this work that we have been doing with the people from the Unifesp, that the University Federal in São Paulo, with Dr. Ana Gades, is in order to identify different resistance that are spread in hospital and also in the environmental. For example, here we identify 2 new plasmids that carry some resistance, the new resistance in distinct species of Acinetobacter, or in Klebsiella pneumoniae that is cause a high mortality rates in Brazil. We also work with the resistance of antibiotics, different strains, with another partners. One of them is a network, Gabriel, that is supported by the Fondation Mérieux in France, and is involved in different cultures spread all over the world, the third countries that we call. So this work especially we have done with the people from the labs in Bangladesh, and we have sequenced the Salmonella typhi. That is the country, because in Bangladesh is a country where the typhoid fever is endemic. So there in the hospital, in Dhaka Children's Hospital, there is a lot of different profiles, resistance regarding some different antibiotics. And we want
ainda não começou.
And we would like to understand which genomics elements that are associated to this resistance. To do that, we have sequenced 70 isolates from Salmonella typhi. We assembled the chromosome in the plasmid, and using this methodology that we have in the lab, we identify new plasmid that carry some resistance. We can see here the resistance that we are able to identify. And the one important thing that we are able to identify some genomics islands that is different from the other one that have been previously described in the literature, and also for variants for this species. We are also working the metagenomics, and we are looking for the metagenomics in different environments. Some impact from the oil. In Brazil, we have a large company for oil, the Petrobras, but we also work in the metagenomics related with health. Here is identifying the virus of flu in different countries, and another metagenomics study. For that, in Sabia, we have developed a part of the Sabia is only to integrate the data from the metagenomics, and we are a special part for the taxonomy of these samples. For example, here is one sample that we have sequenced from the Petrobras company in Brazil, from the sediment of the middle of the ocean here in Bacia de Santos, near Rio de Janeiro, between São Paulo and Rio de Janeiro. And the amount of data that we are able to recover using the metagenomic approach is so huge that we can identify virus, bacteria, and another archaea, and etc. And using different, so Sabia, there is a different models. Some we can identify the diversity of index for this bacteria, and we are able to arrive to the phylum of these genomes in the sample. And we can visualize how many reads we have in each family, in which phylum. Sometimes we can arrive to access the species of this sample. And besides the taxonomy analysis, we are working also with the binding. So we are trying to assemble the genomes for many in this sample. So we start with the FASTQ for the individual sample, and then we put all the samples together and we perform the assemblies of the genomes, and then we start to analyze these genomes. The amount of the genomes that we arrive to from one sample is so huge and so interesting that we can mainly identify new families. That's the work that we are doing now in the sample from this Petrobras. That is Rhizobialis. This is a new genus that we have been described at this moment. We using this methodology, we are able to identify almost the biological physiology of this bacteria. For example, in this new family, we achieved to identify some pathways such as glucose, such as reduction of sulfate and denitrification that has not been described yet. And this is very important for degradation from in sediments in the middle of the ocean. So we are using this applied to the oil gas company. We are using this methodology to identify the microorganisms, to understand the biogeochemical cycles, but also we are working to identify the biotechnological products with the potential for identifying new enzymes, new antibiotics, and also biosurfactants. That is very important. So studying this environment using this methodology is a very important methodology that we are using. Going back to Zika, we are working not only with the virus but also with the humans. So we work with the babies that the babies that has been born with the microcephaly. So last year we published this work where we are able to identify the alteration in the extracellular matrix in the brains of a newborn with congenital Zika syndrome. We start working with the whole genome sequence for the late stillborn babies, and we identify the variants, the rare variants in homozygous and heterozygosis using different database. And then we filter all this variation. We use some different softwares to identify which variants are impact really in the disease. As we can see here, this is the result of the exomes analysis. We identify a lot of important variation, but most of them are in different chromosomes in the human genome. But we identify that in most of the babies that we have sequenced, these are precious samples because nobody has access to this data outside Brazil. We identify a lot of genes related to the collagen family genes that are present in the sample. And then we decide to, with another groups in Brazil, we decide to perform the transcriptome analysis for these samples. And we are able to identify 228 genes that are upregulated and 285 genes that are underregulated. And here we can see that the collagen formation are the genes that are downregulated in the genome, in the transcriptome of these children. And when we perform this pathological analysis of the brain of these kids, these children, we can identify the control. Here in the right is the control. Here is the cell infected. We can see here that in control, we can see here in red, that is the collagen fibers that's present normally in the brain, and in here we cannot see the collagen formation in the brain with the Zika babies. So continue to the Zika, we are working with the infection of the different cells. We have been done with the thymus cells, mosquito cell, astrocyte cells, and other ones, but I will show briefly only the work that we have done with the thymus cells, epithelial thymus cells. So we have the TIMIC, we collaborate this with the Fiocruz Institute also, Foundation, sorry, and then the people there, the researcher there, they have been infected the TIMIC cells, and then we validate the infection using the flow cytometry, and then we extract the RNA to perform the transcriptome analysis. And the analysis of the upregulated genes, we can see here that most of the genes are related to the extracellular structure matrix, the organization of the extracellular matrix, but also genes that are related to the system immune for the people. And we can see that most of the genes are well connected. Here's the connectome for the transcriptome for the genes. And we can see here most of the genes that are related to the organization of the extracellular. And also there are some important genes that link all of this connectome to the genes that are responsible for the response to the virus. When we go in the other side, the downregulated genes, we can see that most of the genes that are downregulated are related to the nucleosome assembly, and most of them are related to the chromatin. Most of they are the histones that are downregulated, and these histones has already been associated to the apoptosis of the cells. And we confirm our results using different approach, and we can see the virus that infected the cells, and also we can see the induction of the infection that changed the extracellular matrix, and we are able to identify that really the thymus can be a reservoir for the virus during the congenital infection. So we are doing the same thing. I have to go fast. We are doing the same thing we have done already. The same thing from the mosquito cells. The results is the paper is submitted, and also for the astrocyte cells. That's the important cell from the central nervous system. And in this case, we are comparing not only the Zika, but since 2012, we have a lot of infection from Chikungunya, Mayaro, Oropouche virus in Brazil. So we are comparing these 4 genes that infect and cause neurological disease in humans. And the last 5 minutes left to me, I would like to talk a little bit about the coronavirus. We started to work with the coronavirus recently. Coronavirus started the nightmare started the beginning of this year that appeared the last of this 2019. It came from China, and they proved that the virus can infect. They quickly infect can see the cytopathic effect in the affected cell. I don't remember which cell else is I don't remember which cells, but they can infect the cell, they can recover the virus, and they assemble, and in January 24, the genome has been published. This is so quickly. So the genomes of the coronavirus is 70% is a polyprotein, WARF1AB, but they have 4 genes, that structural genes, and some of this is a subgenomics importance. Many resource has been created since the beginning of the pandemic in the world. One of them is the NCBI. Of course, China has developed a very nice software interface for analyzing this data. And we have too many also important database and software that we are using in the one is the GSED, that we submit. Most of the sequence has been submitted there, and the next trend that analyzes the data that came from the GSED. And one thing that we start to work with people from all over the world is this phase 4 project. And we want to specify the rules that we have to integrate such many software, many database, and many tools has been developed since the beginning. And so we create a pipeline, a network, the rules that we can for submit for storage to guarantee the quality of the data and to be storing in the same format that the whole community can be used. And here is a picture from the next train showing the COVID in America. And here in red is the samples that came from Brazil, that has been sequenced in Brazil. We have from Chile, Uruguay, Argentina, most of the country in Latin America. And here we can see some diversity when we compare the global genomes that has been submitted in NextTrend and compare it with South America. We can see there is some difference in SNPs that present in the lineage characteristics from the South America. And at LNCC, we started sequencing in March of this year using the mini-ion approach, and we submit the genomes in America. In GSAID, I didn't talk, but in GSAID last week, we have 100,000 genomes deposited for all over the world. But in Brazil, we only have 900 virus at this moment. So it's less than 1% of the amount of the sequence has been deposited for all other countries. So the Ministry of the Ciência e Tecnologia started to this network, the Rede Nacional de Ômicas for the COVID. We are involved in this project, and the main goal of it's involved 10 institutions from the 5 regions of the Brazil. And what we are going to do in this project is to perform the viral genomics, metagenomes, exomes, and transcriptome of the patients and from the virus to identify the spread of the virus in Brazil. We will be using the Centro de Multiprocess, the data from the exome and the transcriptome, and we are using some tools from IA for identifying some patterns. So this is the first work that we performed from SARS-CoV in Brazil with the group from the University of Oxford Imperial College and another partners from Brazil. We start to collect the sequence in the beginning of the spread. The first genome has been sequenced has been identified in February 25. And we start immediately to sequence these genomes. The first state in Brazil, the huge amount was São Paulo, followed by Rio de Janeiro, then Ceará, and the north of the country. So when we start to do this sequencing, we start to collect most of the sample came from São Paulo and Rio de Janeiro, but we collect for other regions of the Brazil. And here we can see this is the city of São Paulo and the Rio de Janeiro, and we can see then the beginning of the spread of the virus, the R rate, that means that the number of the person are infected, can infect another one, is more or less 3. And here in the 16th of March, we start the we started como é que fala? Isolamento, the social is we start the NPIs.
Social isolation.
Social isolation from start in Brazil in 16th of March. So we can see when we start to do this, the government start to do that, some states start to do some isolation with the number of the R rates come to 1, less than 1. But after some day they start to grow, and now we are still in more than 1.5, 1.6 at this moment. And the same thing happened to the city of Rio de Janeiro. So we have sequenced 427 genomes. The quality of the genomes and the assembly is so, the mapping is good. And here we can see that we have more, the sample, the federal states in Brazil that we have samples for all of them. And we try to have genomes, the number of genomes related to the proportion of the infected case at this time. We start to sequence in March and we finish in the end of May. And then is the panorama of our data. So we have some genomes here that we sequence before the first case has been reported. And most of the genomes we have sequenced is before the first death has been supported for all the states in Brazil. And the main linkage in Brazil is the B1.1. And this is characteristic from Brazil. But the one thing that is very interesting in this work that we we are able to identify that the virus arrived in Brazil outside from another countries, mainly Italy. And France is the country that we have imported more virus. But in the beginning, before the Brazil closed the airport, before the Brazil closed the isolation situation, we had already the virus, the Brazilian, I can say that the Brazilian virus are circulating in Brazil before the 16th of March. And then in the second phase, only the Brazilian strain is at the moment. And here we are able to identify that the 3 main clades are reported in Brazil. And one is characteristic from the São Paulo region, another one is from the Ceará, for the northeast of the country, and there is another third clade that we are able to identify. And now we continue to sequencing more virus. We developed, of course, some tools for visualizing. Now we move to use the Illumina. We are sequencing many genomes. We are now looking for the SNPs in the genomes that we are sequenced now. And we can see here that using the Illumina platform, the amount of reads is huge. So it's better than the mini-IO. So we can identify the SNP with the high frequency and with the look frequency. And the platform you can see here, which is the SNPs, the frequency, the number of reads, the deep of reads that we can support that SNP that we are now identifying. So to finish, we develop a lot of database. There are in our web page. We are also performing sequencing and bioinformatics service for the whole community. And I would like to finish to invite everybody to tomorrow. We will have a panel supported by this Interciência, that is in the Federation for the Progress of Science in America. So we'll discuss, we have a panel each month. In each panel, we will have 6 different countries, and we were all talking about the situation of pandemic in America. So I would like to invite you tomorrow at 5 o'clock Brazilian time. We'll have Argentina tomorrow, Colombia, Venezuela, Brazil to talk, and we will have a fruitful discussion about which are the situation, the pandemic in different countries. In October, we will have the second one. So this is our partners, and I would like to thank the agency that support our work. Thank you.
Excellent, Anna. Amazing talk. Congratulations for your work. It's a very comprehensive work, and you work in different areas of bioinformatics. It's very good to have you here and giving your and sharing your experience with us. And now we have 2 questions for you in the questions chat. And the first one is from Erica Souza, and she's asking if she said, I would like to know if Sabiá app is available to use. I went right now to the website, but only the home page is working. The other pages are not available. Your microphone is off.
We can permit the access to Sabiá platform by making an agreement. It's not available for the user of outside.
Okay, then she has to send you an email.
Yeah.
Great. And another one from Maria Rosa Morales. Great presentation. I wonder what are, in your opinion, the biggest challenges to lie ahead in the genome sequencing decoding, and perhaps for the Latin American region. Thanks and regards from Tucumán in Argentina.
Tucumán, Argentina. But where is the question? I don't understand.
Yeah, your opinion. What are the biggest challenges ahead from the genome sequencing decoding, especially for the Latin American region?
Yeah, I think we have to share. We are already done this, but we have to share more information, more database, more facilities to work together, because genomics is so expensive. So we have in Latin America, we have some huge problem at the moment for support science and technology at the moment. So we have to be able to integrate more research and more institutions to work together, and to use the database that are available to make a interesting question to be answered using the database that has been free at the moment. For example, from the sides of the metagenomics, you can use the Tara data. We are working, I have some PhD students that are doing their thesis using the data that are available in Tara Ocean, and is a huge amount of data that's available. So I think we can also work in this side with available data and integrate more softwares.
Great. Thank you. Another one from Vanina Villanova. She said, thank you very much for the inspiring talk. I have a question about the strategy that you are using for minial sequencing for SARS-CoV-2.
We use the Arctic protocols for the minion to SARS-CoV. We are using the primers and the strategy that if you can send me an email I can write my email here. I can send the protocol. It's available in the web, Arctic protocol.
Is this the protocol from the article you published in
Yeah,
Science?
yeah,
Right?
yeah. Right. So you can see there. So it's available. It's in the web.
Okay, great. There is another question from Karen from Colombia. It's in Spanish, but I will translate to English. What is the importance from the use of bioinformatics to study viruses that are very that are organisms that change a lot in the genome? It's difficult to follow the variations in the virus. And she congratulates you for the conference, and ela sauda você from Colombia.
Muchas gracias desde Colombia. I'm not a virologist. I'm working in bioinformatics and genomics, but I think virus is very difficult to work. Fortunately, in SARS-CoV, it's not a virus that we have a lot of mutation. The rate of mutation is not so high, so we can study more easily follow these mutations. But yes, this is important for the influenza virus, that the rate of mutation is so high. So it's important to sequence all the time in order to identify new variation, new mutation that can appear by each year. And I think continue to sequence the SARS-CoV virus is very important, because this virus entering human it has 9 months. So they are changing, they are adapting to the human. So I think we have to continue to sequence the virus to see if there is new mutation, new variation can be affected the drugs and the vaccines. We will still have to sequence a lot of virus, SARS-CoVs.
Another question. Very nice talk about your sequencing services. Are you planning to offer Oxford nanopore sequence as a service using GridION or PromethION?
No, I'm not going to provide service with the mini-ion platform. It's not so. I prefer to use the Illumina.
Only Illumina, great. There is actually a demand from Celeste. She wants to have the links for the pages of the databases you presented. I don't know if it's possible to put the links available. I don't know. Maybe the organizers can do that. And there is another two. Oh,
please.
sorry, Lucy.
And we can share the links with the other participants.
Okay, great, thank you. There are more 3 questions, and then we're going to finish because the time is over. And then I will just ask you 3 more questions. I would like to ask you another question from me, but okay, that's no problem. We can talk later. Thank you for your talk, talking about Salmonella typhi. Did you find resistance to nalidixic acid in the new plasmid PK91 that you found?
Yeah.
Resistance to acid nalidixic.
Yeah, we have already published this paper. So if we put my name in the PubMed, you can find this paper, and all of we have found has been described there.
Okay, great. Sorry, I forgot to say the names of the people that were asking. Sorry for the others. And this is from Augustine Barricala. Many arboviruses are present in the continent for many years and decades, but didn't emerge nowadays. In your experience, what could be the virus or viruses we must expect to emerge in the near future?
We have to study a lot. We have to perform a lot of epidemiological surveillance, genomics, to try to preview the new emergence that will happen. That is normal. That's not a problem that appears. It's not a fake. The virus are there, are in different hosts, and they can enter and in humans. And I think that we have to take a lot of care with our environmental. So if we preserve our environmental, we can't preserve ourselves and the enter of the virus in the city.
And just a comment I'd like to say, especially in the Amazon, there are many viruses that are there, and when you destroy the forest, they can spread to other environments, and that's very dangerous.
Yeah.
Yeah. And then the final one is a comment from Miguelito. Miguelito, he said, congratulations for you and the other bioinformaticians. Thank you, Miguelito. We really appreciate. I hope you're good and stay well.
Yeah.
I'd like to say something, Anna.
Fica bem, Miguel.
Yes. Thank you again.
Thank you, everybody.
Thank
you
you for your very nice presentation. Great work. Congratulations, Anna. Thank you.
Thank you.
And now, thank you.
Thank you, girls.
Thank you, Anna, for being here. It's an honor for us. So thank you very much. It was an amazing talk. Thanks.